Hello, and a very warm welcome to today's Vaccine Insights webinar, entitled Enabling and Advancing mRNA-LNP Medicines from Concept to Clinic. I'm Charlotte Barker, an editor at BioInsights, and joining me today are Lloyd Jeffs, Adam Crow, and Jagbir Singh, who will discuss key insights and experiences that leverage modular microfluidic platform technologies and analytics to enable rapid development, testing, and scale-up of mRNA-LNP vaccines and therapeutics. So now I'd just like to briefly introduce our presenters. Dr. Lloyd Jeffs joined Precision Nanosystems in June 2018. His biopharma services team is responsible for developing and executing custom programs to meet the clinical manufacturing needs of Precision Nanosystems clients. He's a co-author of numerous peer-reviewed publications dealing with the development of lipid nanoparticle therapeutics and is a co-inventor for key patents in the field. Dr. Adam Crow manages a multidisciplinary team at Precision Nanosystems, tasked with developing novel analytical assays related to lipid nanoparticle and nanomaterials for drug delivery. And that team's increasingly leverage cutting-edge technologies such as LC-MS to tackle the complexities of LNP characterization. Dr. Jagbir Singh has 15 years of research experience and has completed over 150 projects in formulation development and manufacturing process scale-up. At Precision Nanosystems, he leads a team of scientists and staff to develop a diverse range of formulations, including mRNA, pDNA, siRNA, peptides, and small molecules. So without further ado, I'll hand over to Jagbir to kick us off with the first presentation.
Hello, everyone. Again, my name is Jagbir Singh, and I'm Director of Preclinical Services at Precision Nanosystems. Again, as mentioned by Charlotte, I lead a team of formulation, analytical, and bioassay specialists to help our customers in their drug development journey. Precision Nanosystems was founded in 2010 to address manufacturing challenges in the development of genomic medicines from lab to clinical scale. Currently, we are a leading end-to-end nanoparticle solution provider with more than 800 instruments installed worldwide. We also offer off-the-shelf reagent kits and clinical lipid library to support your drug product development. In addition, we also provide biopharma services to accelerate our customers' product development from lab to clinic. Our microfluidic nanoassembly technology has been cited in more than 225 publications and protected by various patent applications. So currently, we are also building a biomanufacturing center in Vancouver, Canada, to support your needs for GMP manufacturing. At PNI, we believe genomic medicines are the future. So this was exemplified by the level of response we could achieve by leveraging this technology to develop vaccines against COVID-19. Genomic medicines, they offer that treatment options for diseases arising from both gain-of-function mutations or loss-of-function mutations. So again, we can design payloads construct such as siRNA to silence a gene. We can also design mRNA, pDNA to express proteins of interest in vivo, right? And ultimately, we can also directly repair the gene mutations using library of gene editing tools. So again, the opportunities are vast. It offers a limitless possibilities. So although again, as mentioned, genomic medicines, they hold significant potential for future, there exist a number of challenges in the path of transitioning these therapies from lab to clinic and commercialization. So number one, the inherent nature of the nucleic acid itself, molecules like RNA, they have lower stability and they're prone to degradation in solution. In addition, they are polyanion, negatively charged structures, so they're hard to uptake into the cells. So again, most often they need a kind of a delivery system for transfection of this nucleic acid into the cells. Uh, Again, there remains a significant knowledge gap because, again, this is an emerging technology and it requires a multidisciplinary approach. So, with respect to especially the analytics, which is needed to support formulation development, troubleshooting during the process development. So, there is still a significant knowledge gap which inhibits that transition from lab to clinic. Uh, Genomic medicines, they are complex. They are multi-component drug products where each component needs to come together in a well-defined ratio to ensure safety and efficacy in vivo. So this can make it very, again, difficult to develop and challenging to scale up. Ultimately, like as genomic medicines have potential application in diverse areas, like in treatment of many variety of diseases, so they require different routes of administration, different target issues, again, different application areas. And similarly, like here, it again requires that multidisciplinary team with a wide range of capabilities to come together to deliver on these projects. At PNI, we are helping tackling these problems. First, number one, we are providing our nanosampler microfluidic manufacturing platform for development of these genomic medicines, including the mRNA-LNP based vaccines. Second, we are also developed and developing a library of clinical lipids that can be used by you for encapsulation and delivery of your nucleic acid payloads. So this lipid library is designed for use in development of both vaccines and gene therapy products. And finally, and I would say most importantly, we are also addressing the gaps in knowledge, infrastructure, and experience by supporting our customers through their drug product development journey using our biopharma services. So in next few slides, I'll be taking a little more deeper dive into biopharma services and how we can help you through your journey. This slide presents a snapshot of our capabilities and key functional area we support in your drug development journey. So these areas are formulation development, process development, analytical development, quality and regulatory support, tech transfer, and manufacturing. So in biopharma services, we have an experienced team of about 70 full-time staff supporting these projects. Uh, Among them, at least more than 40 of us has advanced degrees and have been a lot of experience developing these types of drug formulations. So again, we have experience working, taking these projects from discovery to manufacturing to of the clinical batches. To date, we have complete more than 160 projects that are in various stages of development. Formulation development services. So here, like this is like when we're talking about formulation development, that can be divided into three stages. First is the early discovery stage, then more like need for a process scale up, intermediate batch sizes, and then ultimately at scale batches in clean rooms to support your phase 1, phase 2 studies. So here again, in the early discovery stage, we can use our GenY kits or clinical lipid libraries to encapsulate your payload and provide proof of concept formulations in your hand to test your science. We can also develop, like help in development and optimization of your lipid composition and formulation to improve safety, efficacy, and stability. So we can work with our lipids and we can work with your lipids. So ultimately, the aim is to use our microfluidic technology to develop those particles so that we can look at the science together. So again, we can also transfer in your formulations and help in process scale up so that formulations can be prepared from milliliter to liters of scale. Uh, and finally, once we have locked formulation, a locked process, we can tech transfer the formulation for manufacturing on our GMP system to support your safety tox batches and clinical batches. So here, analytical development. So again, analytical development, I would say, is the key to success in any formulation development project. Uh, uh, Again, analytical development, it helps supports in screening, optimization, process scale-up work by providing that insights into changes in drug product profile. Again, we have developed a large array of techniques to understand your formulations better. We have methods for physicochemical characterization of particles as well as individual components such as lipids and RNA. We do routine analysis using particle sizing technique, RNA encapsulation using a dye exclusion assay based on ribogrin reagent, lipid analysis using UHPLC with a CAD detector, ELSD, high resolution mass spec, and other suitable techniques for analysis of lipids, RNA, and other components. In addition, we also offer bioburden, sterility, cryo-TM, residual solvents, and other analysis needed for product release through our partnership with third-party vendors. So again, all in all, we can support both analytical method development and QC testing for release of your products. Finally, I like to say activity is the king. So it is equally important to test the impact of formulation and process conditions on biological activity. In that way, like we can support in vitro screening of formulations during various stages of drug product development to ensure the product retains its activity. So we can perform a majority of standard molecular biology techniques such as automated Western blots, automated ELISA. Uh, we have flow cytometer, RT-qPCR capabilities, and most importantly, again, we have the ability to handle both risk group 2 materials and cytotoxics in our labs. We have also a high throughput microscopy techniques that allows us to evaluate a large number of variables such as dose ranges, antibody ratios, cell types, and so on in relatively short time. So this allows us to establish correct in vitro models early in the drug development journey. So this slide summarizes the type of projects we have been handling in our labs. We have experience in working with a variety of payloads. So yeah, including mRNA, self-amplifying RNA, uh, small interfering RNA, microRNA, circular RNA, other types of RNA, pDNA, gene editing payloads. And in addition to developing this lipid nanoparticle formulations, we do have experience developing other types of nanoparticles. We have projects doing encapsulation of peptides into polymers, encapsulation of peptides into liposomal formulations, or encapsulation against small molecules in other systems like liposomes. In terms of application area, again, a lot of interest since 2020, I would say a lot of interest has come in development of vaccines, both prophylactic and therapeutic vaccines. So we have a lot of experience developing the LNP-based vaccines. More than half of these projects still focus on gene therapy with applications in cardiovascular, lungs, livers, and ophthalmic diseases. And again, we have also started getting a lot of interest to support cell therapy projects. Again, we have experienced most of these products are liquid formulations, parenteral formulations. So we have experienced developing these formulations for administration through variety of routes, including intravenous administrations and intramuscular administration. So in this slide, this is actually my attempt to summarize key milestones from transfer in to preparation of first GMP batch for phase 1 studies. So again, for this timeline, it's the actual timelines is dependent on case-to-case basis, the nature of the project. However, for this is a typical time frame that for a given project. Here, I've taken an assumption that early discovery work has completed. Again, as mentioned, like PNI can help and support in early discovery work. But again, for this timeline, we are taking that assumption a RNA construct, a couple of and lipid compositions are selected, in vitro, in vivo models are selected. In terms of timelines, once a program kickoff happened, it takes 2 to 3 months to get to that formulation lock. Again, it takes another 2 to 3 months for process lock. And while this point, we are also looking at analytical and bioassay method development also. Once we have a locked formulation and locked process, we are ready to transition this formulation to our GMP system for preparation of GLP safety tox batches. And ultimately, the goal is to start preparing the master batch records and defining drug product specifications so that we can continue our journey towards manufacturing of those GMP batches and providing those formulation in your hand to start your phase 1 studies. Again, the activities described in blue box are typically the activities handled by Precision Nanosystems. Some of the activities like sourcing of GMP grade raw materials are performing those in vivo studies, GLP safety tox studies in vivo in non-human primates and mouse and other animals. That is handled by our clients. So again, this timeline is a typical timeline, but this can be shorter, but that is again dependent on the quality and quantity of raw materials. And again, assuming that lead compositions, lead RNA construct, and appropriate biological models are already selected. Finally, I want to highlight that as a Precision Nanosystem is a Danaher life sciences company. So as part of this ecosystem of Danaher Life Sciences company, we are in a position to offer a complete end-to-end solution under one roof. So we can provide you the needed lipid delivery system. We can support you in with your mRNA synthesis needs, and we have a full suite of instruments and consumables needed to make your GMP batches for clinic and commercialization. Thank you for listening, and happy to answer your question during our Q&A session. And now I would like to hand over the presentation to Dr. Adam Crow to take a deep dive into the analytics one should consider during nanoparticle development. Thank you.
Thank you, Jagbir. I'm Adam Crow. I am the Manager of Analytical Development at Precision Nanosystems. And today I'm going to talk a bit about why you might want to go beyond the critical quality attributes to enable and de-risk your vaccine program at PNI. So one important thing I like to highlight with LNPs is that they are an analytically complex drug product. We think of 5 major areas of analysis at PNI. This includes the design of your payload, the quality of the drug substance going in, things such as its integrity, its structure, the composition of the LNP itself. Of course, this is your lipids, your size, polydispersity. Encapsulation, which amounts to where the RNA is located. Is it on the surface? Is it inside? And all of these parameters come back to the idea of potency, which is a central figure. Of course, you can have a beautiful analysis of everything, but if your material is not potent, it loses its ability to be commercialized. Now, today I'm going to talk a bit about the critical quality attributes, or CQAs, and why you may want to go a bit beyond these. So working with many CDMOs, you will get a standard list of CQAs that they will do for your material. In the bottom right here, I've shown a list of what these generally look like. These are things such as appearance, particle size, RNA content, etc. What's important to note is that although these are really important characteristics, they exemplify the minimum specifications for your drug product release. They don't enable a development of the LNP itself. And in fact, you will need a lot more analysis to go from proof of concept to the clinic. To use a rather cliche analogy, we think of this more as the CQAs are the tip of the iceberg. They hide underneath it the ideas about the composition and the design of the LNP, the drug substance and raw material testing, and also a number of assays required to support the development process. In fact, we can count at least 26 different assays that PNI performs to enable the development of LNPs through our commercial services. On the right here, I've indicated a few assays. There are more as well. But as you can see, there is a wide range of assays required to enable a program. So in today's talk, I'm going to give a bit of background on the standard CQAs and give you an indication of why you want to go a step further with those. So first of which, we're going to start with the lipid quantitation. So shown in the bottom right is a pretty typical UHPLC charged aerosol detection chromatogram, identifying the ionizable lipid on the right, two helper lipids, and the pegylated species. Lipid identification is done to make sure your product conforms to its specifications, as well as looking the amount of material there. Can typically be done by charged aerosol detection, evaporative light scattering, or mass spec approaches. And an important factor you get out of this is your RNA to lipid ratio, or N to P ratios, which determine the quality of your product. However, simply analyzing the lipid concentrations in your final material only gives a very limited story. At PNI, we actually take this a step further and do our routine analysis on each of our individual process steps. The reason we do this is exemplified in the right here, whereas you can see that over the sequential process steps, we see a significant drop in the lipid content in process 2. Now, what's important to note here is that although we see a huge drop in the material content, the lipid ratios maintain similar abundances. This is diagnostic for something we usually call aggregation, in which the material aggregates as a whole and is lost. We can actually change process step 2 to account for this and actually recover the amount of material and improve the process. So using analytics to find the problem source here and rectify the issue. Another big area of concern is lipid purity. This is a concern because a lot of the ionizable lipids, in fact, the vast majority, are chemically synthesized by third-party vendors. Many of these vendors that we've worked with have older technologies, such as evaporative light scattering, which tends to overestimate the purity. Now, given on the right here, I've just given an example why this is. So we have both detectors in-house at PNI, and I took an ionizable lipid and spiked in 10% of a known impurity. As you can see by evaporative light scattering, there's less than 1% of relative signal detected, as opposed to charged aerosol, which is almost the stoichiometric amount. This is highly problematic if you are wanting high quality material approaching to the clinic. Now, there are other detector choices, and each one will be specific to a different type of ionizable lipid. But at PNI, we find the charged aerosol detection the most diagnostic. The other real concern with lipid purity is the idea of what other species are present. So shown in the top right here is what was given as a pure ionizable lipid by a vendor with a CQA or COA stating a 95% purity. Now, this was done by ELSD. However, when we take this in-house and look by charged aerosol, we see a number of extra species here amounting to over 20% of the total weight. This is of concern for many reasons. But even from a formulation perspective, this means that the weight of material you're measuring does not actually account for the total amount of ionizable lipid you're weighing out. So of course, your theoretical N to P ratios don't actually match what is truly there. This has impacts on stability, potency, and many other facets. Taking this a step further, we also like to make sure we identify the individual impurities there. So simply knowing that they're impurity is not enough because we need to know what type of impact they have on the formulation. So there are many concerns we have with impurities. There are the obvious ones, such as toxicity concerns, but there are the more subtle ones, which I think are missed quite a bit. Things like the reactivity with the drug substance and changes in the structure. Relating to reactivity, on the bottom right is some internal data we have, which relates to a phenomenon first described by Meredith Packer in 2021. If you see in blue is an mRNA detected by HPLC UV, and in the presence of an ionizable lipid, we see a new species forming, which amounts to a covalent adduct that forms between an ionizable lipid impurity and the mRNA itself, which subsequently inactivates the species, which has severe concerns on the potency of your final drug product. So because of this, at PNI, we make a lot of effort to identify what impurities are present. However, this is a challenging problem. In this example here, which we did with SciEx, we took a model ionizable lipid called MC3 and found two isobaric impurities, both of which show the incorporation of an oxygen atom. The challenge here is that the location of the incorporation of that oxygen atom in the structure has huge implications on its reactivity. So we needed to determine where this oxygen was incorporating. To do this, we use a new technique called electron activated dissociation, enabled by a high-res QTOF instrument from SciEx. In doing so, we're able to fragment every single bond inside the molecule. This allows us unparalleled resolution on the structure. So given in the example here, we can cleave each bond until we see a presence of a double bond. So we can see exactly where structural elements like double bonds and other incorporations occur. Using this approach, we were able to fragment both impurity 1 and impurity 2 and found that impurity 1 contains an epoxification at one of the double bonds, which are really quite generally inert from a formulations perspective. However, impurity 2 showed an N-oxide species in which the oxygen has incorporated at the tertiary amine. These are highly problematic as they are implicated in the adduct formation first described by Meredith Packer. And of course, we want to limit these as much as possible as they will inhibit the ability of the LNP to be manufactured. Switching topics, another common CQA is RNA integrity. Now, RNA integrity is paramount because it is the most predictable impact on the potency of your final material. In that, if you have 50% loss of integrity, 50% decrease, you will lose 50% activity. It's quite that simple. Because of this, you want the highest resolution system to detect integrity. So shown going from left to right is increasing resolution of instrumentation used to look at integrity. And shown is the same mRNA detected through different approaches. As you can see that simple previous approaches such as ion pairing HPLC or bioanalyzer generally way overestimate the integrity of material. At PNI, we use capillary electrophoresis, and as you can see, there are a number of other species that were not being picked up by the other techniques. Because of this, we know that this material has lower integrity than estimated, and this allows us to control reproducibility of incoming material. As mentioned, one of the problems with integrity is that it is directly proportional to potency. This is exemplified in the graph in the middle in red and blue, in which we took a drug substance, a self-amplifying RNA in this case, put it under accelerated degradation conditions to degrade it, and looked at the relative integrity by CE versus the expression of EGFP in cells. As you can see, they correlate almost perfectly. In fact, the R value here is almost 0.9. Therefore, if you have a 10% loss in integrity during your formulation, you've lost 10% of your potency, which is highly problematic. Because of this, we spend a lot of efforts to look at the stability of our drug substance prior to starting formulation processes. We do this by putting the drug substance in a number of different conditions and measuring its relative half-life, as shown in the bottom left here. This allows us to screen conditions such as formulation hold-up times, buffer selections, material interactions, and storage conditions, and allows us to provide that information to our formulation scientists who can incorporate it into their process development plan, thereby cutting off a number of experiments and de-risking the program. Going even further on RNA, that's often overlooked, is the ability to ensure conformity of your drug substance itself. Our RNA is a highly complex molecule with many different facets in it. And if we look at the top left, gives you an idea of all the different types of characteristics that need to be analyzed for reproducible material. Just as an exemplifying one of the facets here we're talking about is this idea of 5' capping. So it turns out that you can have a perfectly intact mRNA that's only missing one methylated nucleotide at the 5' end, and it has, in that case, would have zero activity, as shown in the right. This is very complex to pick up, but in PNI, we use a approach relating to a LCMS to get very careful assessment of the different cap structures of our RNA. This ensures conformity of incoming material and reproducibility of the drug product. Lastly is the area of potency. Now, in my opinion, potency is one of the most underutilized analytical areas in LNP characterization. The reality is that you can have beautiful analytics on every other facet, but if your material it does not show reproducible potency, it'll never go towards the clinic. At PNI, we use potency in-house. We have a full bioassay team with full capabilities, and we use it to support the formulation process. We do this by screening every change in process parameters that we undertake in the development. So exemplified here, this is the same LNP composition processed by 3 different solvent exchange techniques. That's dialysis, centrifugation, and tangential flow. As you can see that using dialysis and then switching to TFF caused a 50-time increase in potency for this material. This allows us to give this feedback to the formulation scientists so that we can choose the best process for individual LNPs and to highlight unseen problems that might not be picked up by CQAs or even other assays. Taking this a step further, we also have the ability to do this in high throughput capabilities. So we can actually measure over 700 samples in a run using automated high throughput microscopy. We can use this to screen things such as lipid selection, lipid ratios, payload designs, processing conditions, and storage buffers, and allows us to get solutions way quicker than you may have doing individual experiments. This has allowed us to move many projects forward at a expedited speed. So with that, I just wanted to conclude some key points from this presentation. And I really want to stress that analytics should not be overlooked when searching for CDMO. They are absolutely paramount to the success of your program. The CQAs are highly important, but they also provide an incomplete picture of what is required for the drug development process. So making sure that there are broad capabilities available is essential when selecting a CDMO. And then finally, that the analytics can de-risk your program by identifying problems early on and preventing unnecessary loss in material. And as a final concluding remark, based on 100+ LNP programs we've been involved with at PNI, I really want to give this idea that performing 2 days of careful analytics can save you weeks of formulation development time. And with that, thank you for being here, and I will pass it over to my colleague, Dr. Lloyd Jeffs. Thank you.
Thank you, Adam and Jagbir, for very insightful presentations. I'm Lloyd Jeffs. I'm the Senior Director of Biopharma Services here at Precision Nanosystems. I want to talk about our microfluidic manufacturing and how that enables the development of genomic medicines, including vaccines. And I'll give an example of a self-amplifying RNA-LNP vaccine that we have developed with the support from the Canadian government. So first off, our microfluidics technology involves time invariant mixing, meaning that we can make formulations less than 1 milliliter up to hundreds of liters of lipid nanoparticles encapsulating nucleic acid payloads, such as messenger RNA, self-amplifying RNA, and other types of RNA, reproducibly, and creating a drug product that is highly uniform. We can do that at different flow rates, starting at around 20 mils per minute on our smaller instruments, and exceeding actually 800 mils per minute currently on our new devices that we're developing, but running typically right now at 200 mils per minute. We can also take these microfluidic mixers and arrange them in parallel to actually scale out manufacturing. And as you see at the bottom of this slide, we are basically controlling the self-assembly of the lipid nanoparticle so that in one step, the nanoparticle is formed where the lipids form around the nucleic acid payload through an electrostatic interaction between the ionizable lipid and the nucleic acid, which is negatively charged. There's other mixing technologies, but they're typically harder to control and to scale. And they typically involve unsteady, turbulent mixing, as you can see in the bottom left of this slide here. So these mixing conditions can constantly change over time and require careful control. Whereas we've solved this for you at Precision Nanosystems with our time invariant mixing and our next-gen microfluidics. As Jagbir mentioned, working with our sister operating companies within Danaher Life Sciences, we offer an end-to-end synergistic solution for developing genomic medicines. So whether it's providing the lipids for making these genomic medicines and the formulations, we can do that. Working internally and with our sister operating company, Aldevron, we can manufacture the plasmid DNA that's required for the in vitro transcription reaction to produce messenger RNA or self-amplifying RNA. Our nanoassembler instruments provide this very precise microfluidic mixing that's scalable. They also contain inline dilution for reducing the proportion of solvent to stabilize nanoparticles. And we can use our downstream processing technologies from Cytiva and from PAL, such as tangential flow filtration, sterile filtration systems, and robotic isolators for fill finish operations as well. So we can do this, as Jagbir said, under one roof. And that allows us to accelerate the development of genomic medicines. So a little bit about our internal self-amplifying LNP program. We received funding at the end of 2020 from the Canadian government through this Canadian Strategic Innovation Fund to develop cost-effective, made in Canada, COVID-19 self-amplifying RNA vaccines. We also received a contribution to build a genomic medicine manufacturing center, which is proceeding with the goal of producing vaccines and other genetic medicines here in Vancouver, Canada. So you may ask, why self-amplifying RNA? Well, in short, it has the potential to be 10 to 100 times more potent than messenger RNA vaccines because the self-amplifying RNA encodes non-structural proteins of the alpha virus that are translated into replicases that make more copies of the self-amplifying RNA. And because you've got a potentially a more potent vaccine, we can reduce the doses by up to a factor of 100, thus reducing the manufacturing burden and allow more vaccine doses to be produced in a shorter period of time. At P&I, working internally, we have actually developed custom vectors that allow us to design plasmid DNAs for self-amplifying RNA and for conventional mRNA. So we've also, as Adam has mentioned, developed analytical technologies to study RNA integrity and identity. And we've been able actually to, in-house, and this is a picture you see on the right, in a local clean room, make tox-grade self-amplifying RNA. So we're quite proud of that achievement, and the analytics look very good as well. So developing a genomic medicine, an RNA lipid nanoparticle genomic medicine, we're focused on, firstly, formulation development using the critical quality attributes. Again, analytical methodologies are essential in doing formulation development right. As Adam has mentioned, we are looking deeper at the quality of our lipids and the quality of the RNA that we're using. And also, the analytics are important for our process development activities, which focuses on the critical process parameters. If you do not have good analytics, you are basically flying blind here. So the better the analytics, the better choices you're going to be making. Also, to save time, using a quality by design or design of experiments approach is very important in basically having a full understanding of the formulation and the drug product that you are developing. So transitioning from discovery to the preclinical stage, our biopharma services team have done this for many, many projects, as Jagpreet has mentioned. These are some of the things that we take a close look at. One is mRNA payload design and the production process by in vitro transcription, or IVT. We look at the analytics or develop suitable analytical methods for the RNA and for the drug product and for the raw materials. For RNA-LNP formation, we're looking again at the composition of the lipid nanoparticle formulation, looking at molar ratios and RNA to lipid ratios or drug to lipid ratios. For particle formation, the microfluidic parameters that we look at are flow rate ratio, total flow rates, and dilution rates. Looking at downstream processing, the choice of buffers is important, and also is the setup of the tangential flow filtration system, which helps concentrate your drug product and perform buffer exchange. And the choice of sterile filters is also quite important as well. Lastly, but not least, is basically the need to develop in vitro and in vivo assays to study the performance of your drug. They're important, and they're also important when you're looking at stability as well, because you eventually need to determine the shelf life for your drug product. So starting stability on as soon as possible is very important. It's also important to look at the stability during the manufacturing process so you can assess if any changes are taking place. So back to our self-amplifying RNA-LNP COVID-19 vaccine that we developed. Here you can see as we scaled using our next-gen microfluidics technology and the instruments you see below, we took two formulations that scaled very well on our smaller instrument, the Ignite, to the intermediate process instrument, the Blaze, to our GMP system, where we saw consistently high encapsulation and consistent particle size and polydispersity. These formulations are made using our proprietary ionizable lipids that we can talk about maybe in the Q&A. For in vivo testing of self-amplifying LNPs, we used a mouse model with a 1 microgram prime, and then after 28 days, a boost. And we measured SARS-CoV-2 specific IgGs by ELISA, looked at cytokine measurements, neutralization assays, isolated splenocytes, and looked also at ex vivo restimulation of the SARS-CoV-2 peptides, and intracellular cytokine staining and cytokine measurements. So we saw for the two formulations, LNP1 and LNP2, they both induced SARS-CoV-2 specific IgG response in mice. We also saw that in vitro previously. Also, both formulations generated neutralizing antibodies against the SARS-CoV-2 virus. And we also saw effective cellular and humoral immune responses. We did not show that data here in this presentation. Moving on to a non-human primate model with the 5 microgram prime and a 5 microgram boost at day 56. We saw significant difference in IgG levels at day 42 compared to the pre-bleeds. And we saw a high response for all the candidates following the prime, as you can see here. And then after the boost, 14 days after the boost, we saw much higher than the other time points, which is typical for a response after a vaccine boost. So summarizing then the RNA development and technology transfer to GMP manufacturing. So really, we've broken this down into 2 parts. One is the preclinical and clinical development of the genomic medicine, where here we are sourcing and qualifying vendors for the production of GMP lipids and API. We're optimizing the process parameters to ensure a robust and reproducible RNA-LNP manufacturing process at the clinical scale. We're qualifying the analytical methods and the bioanalytical methods for the GMP lipids, our API, and our RNA-LNP drug product. We're also developing specifications as soon as we can. I think that's often overlooked, is the documentation, the SOPs, the specifications that you need to develop a drug. And we typically work with, if we have to outsource, say, our GMP lipid, we typically work very closely with our vendor to ensure that we have appropriate specifications so that we can use these lipid raw materials in tox studies and ultimately in human clinical trials. So typically at the end of the preclinical clinical development, we produce GLP tox batches of the genomic medicine and release those internally at PNI. So PNI does not currently have GMP manufacturing capabilities. We hope to have that coming online in 2024. But so what we need to do then is technology transfer the methods, the processes, our materials, and the equipment and all the documentation to a contract manufacturer or another GMP facility. So that's the beginning of our technology transfer. That typically takes a number of months and requires a very detailed technology transfer plan that PNI can help with. At the GMP facility or the CMO, we can do the production of engineering batches, which is, I like to call like a dress rehearsal before the GMP batch, and to ensure everything is working, including the analytical methods, and also to make GMP batches in the clean room that will be used in the clinical trial. That's not the end of it, though, because that starts the QC GMP testing, an extensive review by QA and release, and then packaging and distribution of the finished product. And GMP stability studies and programs will continue for many, many months, typically 2 to 3 years, maybe even longer, for both the GMP drug substance, in this case it would be the self-amplifying RNA, and the GMP drug product, which is the RNA lipid nanoparticle. So we do have a defined set of GMP drug product release tests and core capabilities that you can see in this slide. And this is some data that we have from a long-term storage stability of our PNI vaccine candidate also showing the morphology of it by cryo-TEM. So for this particular batch, we've got up to the 18-month time point at minus 80 degrees, which is the storage condition for this vaccine, where we're seeing it's stable up to 18 months and similar immune responses in the mouse model that we're using. And all the LNPs throughout the study had consistent particle size between 70 and 75 nanometers and maintained high encapsulation efficiency. So to wrap up, the Biopharma Services team at Precision Nanosystems, we've built this over the last few years. This great team, very proud to be working with our multidisciplinary team of scientists and engineers. And we have the capabilities to basically help anyone who wants to develop a genomic medicine, whether it's formulation development, process development, help with analytical method development, or quality control, or quality assurance for that matter as well, and also technology transfer and manufacturing. So thank you very much for your time. I'd like to acknowledge again the Government of Canada for support for our self-amplifying RNA-LNP program, collaborators Dr. Robin Shaddock and Dr. Von Perry and Dr. Matt Stone and the team at SciEx for help with the analytics. And special thanks to Brent Sutherland and the team at the Investigational Drug Program at the BC Cancer Agency. And basically every department at P&I helped us get to where we are today. So there's over 200 of us at P&I, and I think everyone has played a role in basically helping us develop these state-of-the-art biopharma services. So I'd like to thank my colleagues at P&I as well. So again, thank you for listening, and I think we'd be happy to take some questions.